Hi, good afternoon. I'm back with you guys. Just a few topics on glycolysis to wind up that particular topic. So, we're talking about the Pasteur effect. About the Pasteur effect, the inhibition of glycolysis by oxygen, which is an aerobic condition, is known as the Pasteur effect. This effect was discovered by Louis Pasteur more than a century ago while studying fermentation of yeast. He observed that when anaerobic yeast cultures metabolizing yeast were exposed to air, the utilization of glucose decreased by nearly sevenfold. So in aerobic conditions, the levels of glycolytic intermediates from fructose 1,6 biphosphate onwards decreases while the early intermediates accumulate, right? This clearly indicates the Pasteur effect is due to inhibition of the enzyme phosphofructokinase. That could be an MCQ. The inhibitory effect of citrate and ATP produced in the presence of oxygen on phosphofructokinase explains the Pasteur effect. Now, the Crabtree effect, phenomenon of inhibition of oxygen consumption by the addition of glucose to tissues of high aerobic glycolysis, and is known as the Crabtree effect. So basically, this is opposite to that of the Pasteur effect. Why? The Crabtree effect is due to the increased competition of glycolysis for inorganic phosphate. Right? Where in Pasteur effect, it's due to the inhibition of the enzyme phosphofructokinase. So the inhibitory effect of citrate and ATP produced in the presence of oxygen on phosphofructokinase explains the Pasteur effect. Whereas in Crabtree, this is opposite to that of Pasteur effect, and that Crabtree effect is due to the increased competition of glycolysis for inorganic phosphate, Pi and NAD+, which limits the availability for phosphorylation and oxidation, right? And the next topic would be a Rappaport-Lubering cycle. That's Leubering cycle, right? So this is a supplementary pathway to glycolysis which is operative in the erythrocytes of man and other mammals. Rappaport-Lubering cycle is mainly concerned with the synthesis of 2,3-phosphoglycerate, 2,3-BPG in the RBC, 1,3-biphosphoglycerate or 1,3-BPG. Produced in glycolysis is converted to 2,3-BPG by the enzyme 2,3-biphosphoglycerate mutase. So 2,3-biphosphoglycerate is hydrolyzed to 3-phosphoglycerate by biphosphoglycerate phosphatase. So make a note, there's a difference between the usages biphosphate and diphosphate. A biphosphate has two phosphates held separately, 2,3-BPG. In contrast to diphosphate, ADP, where the phosphates are linked together, right? And a diagram to that effect shall be shown here. And it's also believed that biphosphoglycerate mutase is a bifunctional enzyme, and with mutation and phosphatase activity is catalyzed by two different sites present on the same enzyme. And the significance of 2,3-BPG. Production of 2,3-BPG allows the glycolysis to proceed without the synthesis of ATP. This is advantageous to erythrocytes since glycolysis occurs when the need for ATP is minimal. That's a point to note. So Rappaport-Lubering cycle is therefore regarded as a shunt pathway of glycolysis to dissipate or waste the energy not needed by erythrocytes. And the second point is 2,3-BPG, however, is not a waste product in RBC. It combines with hemoglobin, right? HP, and reduces HP affinity with oxygen. Therefore, in the presence of 2,3-BPG, oxyhemoglobin unloads more oxygen to the tissues. Increase in erythrocytes 2,3-BPG is observed in hypoxic conditions, right? And high altitude, fetal tissues, anemic conditions, etc. In all these cases, 2,3-BPG will enhance the supply of oxygen to the tissues. The third aspect would be glycolysis in the erythrocytes is linked with the 2,3-BPG production and oxygen transport. In the deficiency of the enzyme hexokinase, glucose is not phosphorylated, hence the synthesis and concentration of 2,3-BPG are low in RBC. The hemoglobin exhibits high oxygen affinity in hexokinase. Defective patients, hexokinase deficient or defective patients. On the other hand, in the patients with pyruvate kinase deficiency, the level of 2,3-BPG in erythrocytes is high, resulting in low oxygen affinity. Next is conversion of pyruvate to acetyl-CoA. Pyruvate is converted to acetyl-CoA by oxidative decarboxylation. This is a irreversible reaction. Let me repeat, this is an irreversible reaction catalyzed by a multi-enzyme complex known as pyruvate dehydrogenase complex, right? That's PDH, which is found only in the mitochondria. High concentrations of PDH are found in cardiac muscle and kidney. The enzyme PDH requires 5 cofactor enzymes, namely TPP, lipomamide, FAD, coenzyme A, and NAD+. Lipomamide contains lipoic acid linked to lipoamino group of lysine. The overall reaction of PDH is pyruvate plus NAD plus CoA in the presence of PDH gives acetyl-CoA plus CO2 plus NADH plus H+. Reactions of PDH complex. Sequence of reactions brought about by different enzymes of PDH complex in association with the coenzymes is depicted in the figure, which will be shown to you. Pyruvate is decarboxylated to give hydroxyethyl TPP, catalyzed by PDH decarboxylase activity. And dihydrolipoyl transacetylase. Okay, dihydrolipoyl transacetylase brings about the formation of acetyl lipomamide from hydroxyethyl TPP, and then catalyzes the transfer of acetyl group to coenzyme A, right, to produce acetyl-CoA. The cycle is complete when reduced lipomamide is converted to oxidized lipomamide by dihydroxylipoyl dehydrogenase, transferring the reduced equivalents to FAD. FADH2 in turn transfers the reduced or the reducing equivalents to NAD+ to give NADH plus H+, which can pass through the respiratory chain to give 3 ATP, that's 6 ATP from 2 moles of pyruvate formed from glucose by oxidative phosphorylation. The intermediates of PDH catalyzed reactions are not free but bound with the enzyme complex. So in mammals, the PDH complex has an approximate molecular weight of 9 × 10⁶. It contains 60 molecules of dihydrolipoyl transacetylase and about 20 to 30 molecules each of the other 2 enzymes, pyruvate dehydrogenase and dihydrolipoyl dehydrogenase. A comparable enzyme with PDH is aketoglutarate dehydrogenase complex of citric acid cycle, which catalyzes the oxidative decarboxylation of alpha-ketoglutarate to succinyl-CoA. Biochemical importance of PDH. A lack of TPP due to deficiency of thiamine inhibits PDH activity, resulting in the accumulation of pyruvate. In the thiamine-deficient alcoholics, pyruvate is rapidly converted to lactate, resulting in lactic acidosis. Just a second. Okay. Usually after glucose load is observed, PDH activity can be inhibited by arsenic and mercuric ions. This is brought about by binding of these ions with SH groups of lipoic acid. So pyruvate is a key metabolite. Besides its conversion to acetyl-CoA, utilized in a wide range of metabolic reactions, citric acid cycle, fatty acid synthesis, etc. And pyruvate is a good substrate for gluconeogenesis. So that's it about glycolysis. So I'll just get back to you. And now continuing to the next topic would be biological oxidation, right? And oxidation is defined as the loss of electrons and reduction as the gain of electrons. This may be illustrated by the interconversion of ferrous ion Fe2 plus to ferric ion Fe3 plus. Right? The electron loss in the oxidation is accepted by acceptor, which is said to be reduced. Thus, the oxidation reduction is a tightly coupled process. The general principle of oxidation reduction is applicable to biological systems also. The oxidation of NADH to NAD plus coupled with the reduction FMN plus FMNH2 is illustrated. In the above illustration, there are two redox pairs. So the illustration is NADH H plus NADH plus hydrogen. Okay, is reduced to NAD plus. And it also releases two hydrogen atoms plus two electrons, leading to the formation of FMN, which gets converted to FMNH2. Right? The redox pairs would differ in the tendency to lose or gain electrons. Redox potential is E0. The oxidation reduction potential, or simply redox potential, is a quantitative measure of the tendency of a redox pair to lose or gain electrons. Redox pairs are assigned specific standard redox potential, E0 volts, at pH 7 and 25. The redox potentials of some biologically important redox systems are given in Table 3. The more redox potential represents a great tendency of reductant to lose electrons. On the other hand, a more positive redox potential indicates a greater tendency of oxidant to accept electrons. The electrons flow from a redox pair with more negative E0, that is, redox potential, right, to another redox pair with more positive E. The redox potential E0 is directly related to the change in the free energy. And if you see the E0 volts for various oxidation reduction systems would be succinate or alpha-ketoglutarate has minus 0.67, 2 hydrogen plus or hydrogen minus 0.42. NAD plus NADH is minus 0.32. NADPH plus NADPH is minus 0.32. Lipoate oxygen reduction is minus 0.29. FAD and FADH2 is minus 0.22. Pyruvate or lactate is minus 0.19. And fumarate and succinate is minus 0.03. And cytochrome B is with the Fe3 plus or Fe2 plus would be minus 0.07. Coenzyme Q would be plus 0.10. Cytochrome C1 would be plus 0.23. Cytochrome 3C would be 0.25, and that's all. That's for plus. Cytochrome A would be plus 0.29, and half of oxygen or water molecule would be plus 0.82. So these are all the standard redox potential of some oxidation reduction systems. And the electron transport chain. The electron-rich carbohydrates, particularly glucose, fatty acids, and amino acids, undergo a series of metabolic reactions and finally get oxidized to CO2 and H2O. The reducing equivalents from various metabolic intermediates are transferred to coenzymes NAD plus and FAD to produce, respectively, FADH2 and NADH. The latter to reduce coenzymes pass through the electron transport chain ETC or respiratory chain, finally release oxygen to water. And the next topic would be the mitochondria and the powerhouses of cells. And just before that, I'll just give you an overview of biological oxidation EDC electron transport chain. The passage of electrons through the EDC is associated with the loss of energy. A part of this free energy is utilized to generate ADP from ADP. Now, in mitochondria, as being the powerhouses of cells, the mitochondria are the centers for metabolic oxidative reactions, which are the centers of metabolic oxidative reactions to generate reduced coenzymes NADH and FADH2, which in turn are utilized in the EDC to liberate energy in the form of ATP. For this reason, mitochondrion is appropriately regarded as a powerhouse of the cell.